Yeah, hi guys. So today we are going to discuss another concept in the 100 concept session. Yeah, today the concept is urine formation, which is elimination of nitrogenous waste. Okay, that is the topic in the human physiology for NEET. Okay, guys, this is also most important topic for most of the aspirants. They don't understand deep analysis. This is the topic which is about understanding level questions they can ask you. Of course, there are in the physiology they can ask you understanding level questions, observation level questions. Observation level questions, they might be focus on the diagrams and what is going on, and application oriented questions. If the application oriented questions in the physiology, they can ask you the diseases and medicines like that. What is the reason for the disease? And yeah, application, understanding, observation like questions they can ask you. This is understanding level, mean how the urine formation. Okay, urine is what? Urine which is nitrogenous waste. If it is accumulated in the animal tissue, that tissue going to be toxic. Yeah, so that should be eliminated. There is a process called urine like excretory system. Okay, urine formation mainly involves 3 steps, guys. 3 steps, that is glomerular filtration, tubular reabsorption, tubular secretion. These 3 steps involved in the urine formation. Okay, yeah. Before that, what is glomerular? Yeah, it is part of nephron. Yeah, 3 steps. How many are there? No one still, because of it was late class started. Yeah, urine formation. What is this? Can I bring another one? Sir, how did it write?
From that one?
No, it can be damaged.
Now, what I should do is here.
ఇది డ్యామేజ్ అయింది ఉంది ఆ రూమ్లో, బోర్డు ఖరాబ్ అవుతుంది. ఇది లేదు, ఇది టిప్ లేదు, థర్డ్ రూమ్. Yeah, urine formation mainly 3 steps. Okay, first one is glomerular filtration. Second one is tubular secretion, and third one is tubular reabsorption. Okay, so what is glomerular? First, you come to know what is glomerular. What is glomerular? What is tubular secretion? What is tubular reabsorption? Tubular is what? Okay, so in the kidney, this is the kidney. Kidney has mainly 2 parts.
Cortex, medulla.
Yeah, cortex and medulla. These are conical shaped parts, medullary pyramids, which is 12 to 15 are there in each kidney. Okay, the cortex region, which is support for the this is the blood vessel, which is renal artery. blood vessel which is renal artery, which is enter in the pelvis region into this middle part, the cortex region which is between the medullary pyramid, the renal artery which is enter, and this renal artery further divided into arteriole, arteriole further divided into capillary, capillary further divided into again arteriole. Yeah, efferent arteriole and efferent arteriole, efferent and efferent arteriole. So this is renal artery part, means circulatory part is turned into the glomerular, circulatory part turned into the glomerular. Just yeah, this is artery, the blood vessel. Yeah, I'll write it here. Renal artery, renal artery, artery further divided into arteriole, right? It can be carry, renal artery carry the blood from where?
Body to kidneys.
From the heart
To kidneys.
Heart, the left ventricle. From the left ventricle, they can carry the blood. How much? 1/5 of the blood. 1/5 of the blood from the heart to renal artery carry into the kidney. First artery, artery further divided into
Renal. Hepatic.
artery further divided into arteriole.
Renal artery divided into
Arteriole. This is the arteriole we can call efferent arteriole. Efferent arteriole. Right? This is the arteriole further divided into capillaries. Okay? Yeah. This is arteriole further divided into capillary network. Capillary network. This is the capillary network we can call glomerular. Glomerular. Okay? This glomerular further there is again to end with arteriole. This is efferent arteriole. Efferent arteriole. So the blood they can carry, this is the blood, the combination of urea and oxygen. They can carry urea and oxygen. So urea can be filtered, not oxygen filtered. Because of oxygen they can give entire nephron, the oxygen will supply. So oxygen work not yet done here in the glomerular. Urea, it is done in the glomerular itself. Urea, they do not pass here in the efferent arteriole. This glomerular part, it is filtered for the urea only. Got it? Yes. So there is filter tube. Filter tube. This is filter tube we can call Bowman's capsule. Okay? So this is nephron is located here, Bowman's capsule like this in the medullary pyramid. There is a previous year question, which part of the nephron is located in the medullary pyramid? loop of Henle and collecting duct also. Okay, previous year NEET question. So guys, the first step is glomerular filtration. The filter the 1/5 of the blood, mean it is about 1100 to 1200 ml of blood they can carry and filter. Okay, so per day, which is 180 liters per day. Per day 180 liters. Okay, per minute 125 ml per minute. This is glomerular filtration rate. Okay, glomerular filtration rate is what? The rate at which
primary urine is formed.
The rate at which glomerular filtrate filtrate is being formed.
In Bowman's capsule.
Yeah, being formed enough. Okay, the filtrate at which the glomerular filtrate is being formed. Okay, yeah, how much? Which is 125 ml. Yeah, that is actually filtration is going to take place. What kind of elements? Water they can filter, ions they can filter,
All liquid part.
and urea, such a like all liquids they can filter here.
Cellular water maybe.
Yeah, that is the first step of glomerular filtration. The glomerular filtration for day it is 180 liters, for minute it is 125 ml. Is per nephron or per kidney?
Per nephron.
For nephron 180 liters.
Per kidney 180 liters.
Yeah, per two kidneys. Two kidneys as how many nephrons?
Million.
1 million. Mean 2 million nephrons are there. 2 million nephrons together to form 180 liters per day or 125 ml per minute they can being formed this solution. Filtration is going to take place. Yeah. This filtration is crossed 3 main layers. This filtration is crossed 3 main layers. What are the layers? This is the question. This filtration is caused by 3 main layers. What are the layers? Here, this part. What are the layers? This is all our Malpighian body. Okay, one urinary tube, one is glomerular. Mean this is circulatory system, this is urinary system. Together we can call Malpighian body.
Malpighian capsule.
Yeah, Malpighian capsule. Okay, if come to the, it is Bowman's capsule. This is Malpighian capsule. Okay, what are the 3 layers we are going to discuss here? Okay. Like 3 layers mean this is the blood vessel, capillary network, okay, this is the capillary, this is the Bowman's capsule, Bowman's capsule. Capillary, blood capillary is made up of with, Chinna, tell me, blood capillary made up of with,
Single layer of
yeah,
epithelial cells.
yeah, single layer of epithelial cells, blood capillary made up of with this capillary made up of with single layer of epithelial cells called squamous epithelial cells. Squamous epithelial cells in the blood capillary we can call endothelial cells, okay, endothelial cells. Endothelial cells. These endothelial cells in the glomerular, there is a pores, open pores are there. These pores we can call fenestra. Fenestra we can call fenestra. And this Bowman's capsule, which is the Bowman's capsule walls made up of with, Bowman's capsule walls are made up of with which type of epithelial cells? Squamous epithelial. These guys, these cells, squamous epithelial cells. Okay. Squamous epithelial cells. Squamous epithelial cells. These squamous epithelial cells, they can directly contact with this glomerular, directly contact with this glomerular. They can modify it into finger-like structures. Squamous epithelial cells, they can modify it into finger-like structure. These structures we can call podocytes. Podocytes. Okay? Yeah. Epithelial cells, the functions of the epithelial cells, epithelial tissue is filtration, okay, secretion, absorption, secretion, diffusion, okay, the functions of the epithelial tissue. Here there is a filtration. The function of this epithelial cells is filtration. Squamous epithelial cells is looking like this squamous epithelial tissue. This is the squamous epithelial tissue which is looking like this, and these tissues has their own base membrane. Yeah, this is own basement membrane. So, any epithelial tissue, there is a cells and base membrane. Base membrane, okay? Epithelial cells has its own cells and base membrane as well, okay? If come to this glomerular, there is a endothelial cells, and there is a squamous epithelial cells in the Bowman's capsule. Between them there is a base membrane. So how many layers crossed here? 3 main layers. What are they? One is one is the endothelial cells, endothelial and podocytes of the squamous epithelial cells. Yeah, podocytes of Bowman's capsule, and third one is base membrane. Base membrane. These 3 layers they can cross. What about base membrane? Base membrane is acellular, cellular. Base membrane is what? Here I am going to write base membrane. Base membrane is acellular or cellular? Acellular. Okay. Base membrane is acellular membrane, mean non-cellular membrane. Mean non-vascularized. Okay, they can take the nutrition from the blood through, there is a process called diffusion. Okay, they can take the nutrient from the diffusion itself. So how many layers itself cross in the urine filtration? Initially in the glomerular filtration are 3 layers, that is the endothelial cells of the capsule, that is capillary network, which is glomerular, and the squamous epithelial cells of the Bowman's capsule, which is we can call podocytes. There is filters slits are there in the glomerular called fenestra. Between them there is a basement membrane, that is 3 layers they can crossed in the glomerular filtration per minute, which is 125 ml, okay, and per day, which is 180 ml. This is the filtration rate of the glomerular. Okay, I hope you guys understand this. Okay, yes. And next part is tubular secretion, right? Tubular secretion. Okay, you can call tubular absorption, reabsorption. Second step we have, tubular reabsorption. Okay, tubular reabsorption. What is tube? Which is the tube which is peritubular capillary network around the nephron. Okay, yes. Yeah. Okay. This is it appearing? Okay. Okay, this is the peritubular network around the nephron. Actually, there is 90% filtration is going to take place, 70 to 80% filtration is going to take place at the part of PCT, right?
Reabsorption.
Yeah. So, which is about the matter of 180 liters per day, okay, which is they can carry forward into the Henle loop and DCT part and collecting duct finally. So, which is 70 to 80% reabsorption is going to take place in the part of PCT, okay, where from this blood vessel to PCT part, from the blood vessel to PCT part. No, reabsorption is going to take place from the renal tubule to blood vessel. From the renal tubule to blood vessel in the PCT part, 70 to 80% reabsorption going to take place, okay, from the renal tubule to capillary network, okay, reabsorption. From renal tubule to capillary network. Because there are filtrates are going to take place, which is urea and glucose, other minerals. Yeah, there are like certain minerals which is potassium, sodium, okay, yeah, bicarbonates, okay, hydrogen ions. These are, they can again to reabsorption into the capillary network. This is the second step, okay. Third step, tubular secretion. This is, yeah, from the tubes, capillary network into the renal tubule, there is a secretion is going to take place. The remain the wastage which is finally secrete into the renal tubule, okay. These are three steps, okay. Yeah, we are going to discuss the detailed manner about the glomerular filtration, tubular reabsorption and tubular secretion. These are all over comes under urine formation, okay. Yes, we are going to discuss about the glomerular filtration first. Yeah, mechanism of urine formation, which is glomerular capillary network. Yeah, glomerular filtration. Yeah, first it is the first step, and the filtration of blood which is carried out the glomerular, and it is called glomerular filtration. The average which is 1100 to 1200 ml, 1/5 of the blood carry forward by the renal artery, okay. Yeah, the blood is filtered by the kidneys per minute. It is roughly which is 1/5 of the blood, okay. Glomerular capillary network which is cross 3 layers. We discussed endothelium of the glomerular blood vessels and epithelium of Bowman's capsule, which is squamous epithelium, and basement membrane as well, okay. Yeah, yeah. What is filtration rate? What is filtration rate? The rate at which the glomerular filtrate is being formed per minute, which is 125 ml, okay. Ultrafiltration, we are going to discuss now ultrafiltration in the glomerular filtration. In the glomerular filtration, we are going to discuss ultrafiltration, okay. Ultrafiltration, there are steps. The filtration under pressure, ultrafiltration, the filtration under pressure, okay. That is GPHP, CHP and BCOP. What is GPHP? What is CHP? What is BCOP? We are going to discuss. GBHP, glomerular blood hydrostatic pressure, and CHP is the capsular hydrostatic pressure, and BCOP is blood colloidal osmotic pressure, Okay, we are going to discuss about it in the glomerular filtration, which is ultrafiltration. Okay, yes. Yeah, I'm gonna take here only. Yeah, first GBHP, glomerular blood hydrostatic pressure. Blood hydrostatic pressure is what? What is the structural formation of the glomerular here? Okay, yeah. Okay, this is efferent arteriole. Okay, this is efferent arteriole. Efferent arteriole and efferent arteriole. The diameter size of the efferent arteriole is higher than efferent arteriole. Okay, the size of the diameter which is the higher than efferent arteriole size is higher. So, if the blood pass like 1100 to 1200 ml of blood if pass into this blood vessel, okay, there is the diameter of size efferent arteriole created pressure here that leads to the filtration is being formed here 125 ml per minute, which is formed filtrate because of this pressure, which is created by glomerular blood hydrostatic pressure. Glomerular blood hydrostatic pressure. Okay, yeah. So, which is how much it is? 60 mmHg. 60 mmHg. which is GBHP. Glomerular blood hydrostatic pressure. Okay, yeah. After that, there is another pressure which is located here, which is CHP, capsular hydrostatic pressure. Capsular hydrostatic pressure. Here capsular is what? Here what is capsule? Bowman's capsule. Bowman's capsule. This is also have pressure. How it is going to create pressure? Mean if the solution is enter into the Bowman's capsule, if it is filled, the solution if it is filled, okay, this is the solution. Yeah, this solution is created here. This is the liquid. Solution nothing but there is a filtration, that is like glucose or urea, this is like fluid, okay, all over fluid which is enter into the Bowman's capsule. This is the fluid as like hydrostatic pressure. Hydro mean it is a liquid. This liquid is create opposite direction pressure which is filtrate, right? this is filtrate, right? Filtrate into this place. This place also created the pressure opposite direction. They are stopping being the filtrate. There are certain amount of pressure, they can stop the filtration here. Okay, it is also created opposite direction pressure. Opposite direction pressure. This capsular fluid, they can create opposite direction pressure. Okay, this is about up to 20 mm Hg. Up to 20 mm Hg. Capsular hydrostatic pressure, which is created opposite direction of glomerular filtration. Okay, that is 20 mm Hg. Of course, this is the blood has certain nutrients like this is the blood vessel. As RBCs they have blood, as proteins also there is a globulin proteins, okay, proteins and other ions. Yeah, albumins like proteins, okay, albumins like proteins are there. Yeah, albumin. These proteins and these nutrients, they do not allow the water filter here. They do not allow water filter here. Okay, these guys, they do not allow the filter here because of they can hold the water. They can hold the water, that is colloidal in nature. These albumins like proteins are there in the blood, like other nutrients like glucose, okay, these kind of minerals also they can hold the water which is being formed filtrate here. That is blood colloidal osmotic pressure. These guys we can call osmolytes. Osmolytes. Osmolytes, what are the osmolytes here? That is globulin proteins. Globulin proteins and glucose. Glucose, these they can attract the water. They can attract the water, other minerals also. They can attract the water opposite direction. They can attract the water opposite direction. They do not allow the filtration. This is also one of the pressure which is we can call 30 mm Hg. So there are three forces involved in the glomerular filtration, ultrafiltration. What are the forces? First is GBHB, glomerular blood hydrostatic pressure. Second one is capsular hydrostatic pressure. Third one is blood colloidal osmotic pressure. First one is based on the blood pressure, which is the like structurally changes of efferent arteriole size, that will create pressure here for the filtration. And second one is there is a capsular network which is capsule, okay, Bowman's capsule has certain fluid which is they can directly like against the filtrate, okay, they can pressure created against the filtrate. And third one is capsular blood colloidal osmotic pressure. This is the blood has such elements like globular proteins and carbohydrates like glucose and other minerals. They can hold the water for the which is filtrate, okay, they can create against pressure for the filtration. So here just look into here. Yeah, net filtration rate is what? Net filtration rate. Net filtration rate mean there is a GBHP minus CHP plus BCOP. GBHP minus CHP plus BCOP. GBHP which is 60 mmHg minus 20 mmHg plus 30 mmHg mean 60 mmHg minus 50 mmHg. 30 plus 20 is 50.
10
60 minus 50 is 10 mmHg is the net filtration rate, that is 125 ml per minute. 10 mmHg create 125 ml. They can ask you the question, if there is any no pressure against this, there is no CHP and there is no CHP, if there is no BCOP, if there is, yeah. So if it is 60 mmHg, 60 mmHg into, this is about, if the 10 mmHg is 10 mmHg, which is 125, which is 125 ml. If it is 60 mmHg, here's how much. They can ask you in the exam. Of course, there is no against pressure is created, which is capsular hydrostatic pressure, and blood colloidal osmotic pressure is not there. Per minute, how much they can filtrate? 750 ml. Yeah, if it is 10, 125. If it is 60, yeah, 750 ml. Yeah, you are great. Okay, so 750 ml up to the 750 ml, they can carry forward if there is no hydrostatic pressure, no like colloidal hydrostatic pressure. Okay, yeah. Of course, they don't ask you if they want to make the question like difficulty. Okay, yeah, there is a chance. Yeah, it's 6. Yeah, guys, today topic is over. Tomorrow we will going to discuss the detailed discussion of this filtration rate, and there is a tubular reabsorption and a tubular secretion we are going to discuss tomorrow. Thank you.